Thanks very much to EID for fantastic meeting and also asking me to speak at this symposium on what I think is really quite an interesting area of virology that's emerged in the last year or two. It's a silly title, obviously it's more than a coding sequence. RNA viruses are actually extraordinarily complex, and I'm going to just go through some of the background and intricacies of their replication and evolution in the next couple of slides. So they're very diverse. Their defining feature is they use RNA as their genetic material instead of DNA, and of course this certainly makes this whole field somewhat unfamiliar to virtually everyone else working in biology. Uniquely, they obviously have to have an RNA-dependent RNA polymerase to replicate their genomes, and to an extent these are somewhat homologous between the wide variety of different RNA viruses that have been found so far. They're kind of diverse in the sense that they not only have this weird RNA as their genetic material, but they can be single or double-stranded in terms of their genomes. If they're single-stranded, the genome can be in the sense orientation, which means it directly codes, or it can be antisense, so it has to copy first before it makes a protein. And some viruses actually manage to have genomes where some segments are coding and some are antisense. I'm not going to show a tree of all RNA viruses. In fact, you can't really link them all together, but suffice it to say there are 50 or more virus families into which RNA viruses are currently classified. They infect mammals, and that's sort of what I work on, but they also infect plants, fungi, bacteria, and so on. So they are in all domains of life, and they have their own very intricate and subtle evolutionary histories. Their genomes are compact, and the reason for this is that the RNA-dependent polymerase that copies their genome is non-proofreading, and there seems to be some sort of fundamental limit on how big a genome can get before it incorporates too many errors to make it non-viable. So RNA viruses in general range from about 7,000 to 15,000 bases in length, with one or two exceptions. And here's a sort of typical genome. It's compact. Here are the genes encoding replication enzymes, the polymerase is here. Here's a helicase and protease here. And this red area here are all the genes that encode structural proteins, capsid and envelope. And in that, also, there has to be a whole load of replication elements that govern the way in which the RNA genome is transcribed and copied, and also how it directs translation. And hepatitis C, like many other viruses, has a different translation initiating mechanism for other most cytoplasmic components. In fact, this iris, or internal ribosome lymphocyte, is a very complex RNA structure that is involved in direct binding of ribosomes internally onto the RNA genome, so it can actually initiate translation from way inside the RNA sequence rather than scanning from the ends. And this gives RNA viruses many advantages in terms of host control. So that's the sort of background. They are ridiculously compact and complicated to work on, even though they seem at the outset very simple. There are other peculiar attributes that they have, and one of these is composition. And I'm just going to take you through a little bit of background to dinucleotides, okay, which has been a very interesting recent area of research. So dinucleotides are pairs of bases, so it really refers to, for example, C followed by G is the conventional CpG dinucleotide that you have heard about. They're important in vertebrate evolution because CpGs are targets of methylation, and they tend to be lost. So vertebrate genomes have a profound suppression of CpG and actually UpA dinucleotide frequencies compared to the expected frequency based on their GC content. Remarkably, even though RNA genomes do not even really enter the nucleus, and they're certainly not subjected to that type of methylation process, they are also consistently underrepresented in their genomes. And just taking a big survey through mammalian RNA viruses, most classes of RNA viruses actually show profound suppression of CpGs. This y-axis scale shows 0 to 1, where 1 is the frequency we expect by chance based on their GC content, which is shown on the x-axis. And you'll see figures of around 40-50% representation compared to the expectation based on the mononucleotide composition. The same is also true for plants. So plant viruses profoundly suppress CpGs, but it's not true for insect viruses. And of course, one of the sort of clues here is that it's plants and animals that typically suppress, vertebrates typically suppress CpGs in their genomes, and the viruses that infect them also suppress them as well. So there have been many theories as to how this actually works, and of course they tend to focus on the idea that RNA viruses are suppressing CpGs to really fit in better with the cytoplasm which they're expected to translate their genomes and replicate. Although what's driving this selection is really very unclear. Many studies have, without really any evidence, decided that perhaps this suppression is some form of avoidance of host non-host recognition mechanisms, so that perhaps RNAs with high CpGs, which perhaps originate from a different organism entirely, might be recognized as foreign because they have too many CpGs. Although how that actually works mechanistically isn't really entirely clear. So the project really started with an investigation of really discovering whether CpG and perhaps other dinucleotides have a direct effect on viral fitness. And the only real background to this type of work was really an accidental discovery made a few years ago that two groups actually who were trying to make an attenuated poliovirus vaccine manipulated the poliovirus genome to codon de-optimize it. Okay, so they picked all the unfavored codons to really turn down the translation rate of the genome. And to their surprise, they both found massive attenuation of replication of the polio mutant, and of course that's good in terms of making a vaccine. But their explanation for why it didn't replicate properly was wrong. In fact, Burns had actually, from CDC, actually published a paper subsequent to these initial studies, showing that actually it was the fitness attenuation that was observed in these mutants was really just the result of accidentally increasing the number of CpGs and UpAs in the genomes, although the mechanism for that wasn't really uncovered at that time. So that was intriguing, and it's something we wanted to try it in our own test systems. And also to take the other sort of argument is if you can attenuate replication by increasing frequencies, what's the effect of actually artificially lowering them? And if you go back to the previous slide, they are suppressed, but they're not zero. And in fact, CpG can be made to be zero if you want it to. And then secondly, if there are these effects, then how is the abnormal composition of RNA viruses recognized? Are there new pattern recognition receptors that signal the presence of high CpG or UpA? So our model virus was Echo 7. That's what we started with. I wish we hadn't, but there you go. It's a simple RNA virus, supposedly. This is the open reading frame with the structural genes. This is a non-envelope virus. Infects the gut. Here, non-structural proteins with the polymerase down at that end. So some similarities with hepatitis C. We picked it because a lot is known about the genome of the virus, and there's a reverse genetics model, so we can actually make DNA copies of this, mutate it as we like, make RNA from it, and actually recover infectious virus from the RNA. There are things that are known about it, like hepatitis C has a big 5' UTR structure that we wanted to avoid in our mutagenesis. There's a 3' UTR, and there's a replication element in the middle. This is just a plot of synonymous variability, so it's profoundly suppressed here. This is involved in replication initiation. Other parts of the genome, however, seem to be relatively unstructured and ripe for a bit of mutagenesis, and these are the two regions that we actually picked, although we've since done far more extensive mutagenesis of this genome. And what we really wanted to do was to take these two regions here and make synthetic DNA sequences where we've actually altered the frequencies of various dinucleotides, and the ones we picked were CpG and UpA. This is the frequencies of those two dinucleotides in region 1. There's a similar table for region 2, which I won't show you. This is the native frequency on that y-axis scale I showed you earlier on. So there is some suppression of CpGs in the native sequence and also of UpA compared to expectations from the overall composition. We made a permuted control, and this is an essential internal control for the experiments where we change the order of codons but keep the dinucleotide frequencies the same, which isn't actually very easy. bioinformatically. But essentially the idea is to demonstrate that if you just simply mess up the sequence but keep everything else the same, you don't impair replication of the virus. So that's necessary first to demonstrate that the system works. We can then actually make these insert sequences into those regions where we can take out all the CpGs, for example. There are 51 in region 1. We can get a residual N frequency of 0 while keeping the coding the same, and that's because there are many ways of encoding arginine. We can take out most of the UpAs as well if we want to. Unfortunately, there is a few tyrosines that remain in that part of the genome that we can't take away. The other thing we can do is artificially increase these, and so we can add in 129 CpGs or 109 UpAs. So the first basic experiment was to construct these mutants, and here's a whole array of mutants in region 1 and region 2, wild type, permuted, and then high CpG in region 1, region 2, or both, high UpA in both, and then low mutants, so low CpG and low UpA in both. I'm using this sort of coding, so capital letters mean high, little letters mean low, and P means permuted. So here's a multi-step growth curve of where we try to just biologically characterize the mutants we've made. So Echo7 grows like mad in the lab, and even as a rather impractical virologist myself, we could actually get this system to work quite well, though we had a lot of help from Dave Evans, who's a long-term collaborator in University of Warwick. So we made mutants, we made RNA, and we infected cells in a multiplicity of 0.01, so it had to go through 2 replication cycles, and we see this nice prolonged log phase of replication of wild type, and this is the permutant mutants, and essentially these replicate the same. There are error bars on this, but they're too small to see, really. So we were satisfied that just changing codon order doesn't make a difference. There's nothing apart from the composition in these 2 regions. And this is the result that we sort of expected to see. So if we increase the number of UpAs in the region 1 and region 2, we got about a log attenuation, so it just grew slower than wild type, although obviously it was still viable. And this is really quite dramatic. This is the high CpG mutant, where in fact we can reduce replication by up to 4 logs compared to wild type. And then remarkably, and this is work that we've really confirmed in other systems as well, if we take CpGs and UpAs out, that in the exponential period of replication, it's showing about a 1 log advantage over wild type. And in fact, we've since made mutants where we've replaced 70% of the genome with low CpG and UpA, and these actually replicate even faster than that mutant there. So it's really quite a remarkable finding, and of course it raises a whole series of evolutionary questions about why CpGs and UpAs are at the level they are. Why on earth don't they just take them out and actually replicate faster? So we've done some other phenotypic work, Plaque assays, for example. You can set up monolayers and then infect them with virus and actually look at the size of plaques after a fixed period of time. So here's the wild type, normalized to wild type mutants, very similar. High mutants are much lower, very small plaques compared to wild type, but actually the low CpG UpA viruses generated larger plaques than wild type. And a key measure that I'll come back to later on is RNA to infectivity ratio. This is the number of RNA copies you need in a virus stock to make an infectious unit. And for wild type and permuted, you need about 300 RNA copies or 300 virions probably to make 1 infectious center. But if you look at the high mutants, you need about 1,000 times more virions to generate 1 infectious center. And about 100 times more for the high UpA. So biologically, these are really quite different in terms of replication. We wanted to confirm the better replication ability of the low mutants, and we did a whole series of competition assays where we co-infected cells at the same time and differentiated them by restriction enzymes after a number of passages. So here's wild type and permuted, and after passage 15, these were still equimolar in amounts. But if we tried any of the low mutants against wild type, these immediately outcompeted the wild type. So even after passage 5, we're seeing a substantial fitness advantage of the low mutants. I'm not going to go into this in any detail, but essentially we did a whole series of cross-competition experiments to just really confirm this. And the important bit is the bottom. This is a sort of fitness ranking based on these cross competitions, where wild type and permuted were the least fit of all the viruses, whereas the double low mutant at the end here was the most fit. And the fitness gain is proportional to the number of CpGs and UpAs you take out. This phenomenon extends to other systems as well. Dave Evans, many years ago, made a Replicon for Echo 7. And a Replicon is a system where the structural genes are replaced by a reporter gene. In this case, it's luciferase. And the idea then is you can study replication without the virus having to assemble and affect new cells. Dave Evans was always very happy with this. This shows a period of post-transfection RNA level or luminescence from this reporter gene over time, and you see that actually exponentially rises and steadies off. We did, however, discuss on the phone and then noticed that actually the luciferase gene itself was derived from an insect, which has an extremely inappropriate frequency of CpGs compared to mammalian sequences. It's about 1.1. And so the best thing to try was to then take the CpGs out of that and then re-express the replicon. And remarkably, and I think Dave was quite surprised, having worked on this for many years, that actually there's a fundamental replication defect in it. And by simply taking these nucleotides out, you can actually enhance replication by about sort of 50-fold over time. And if you take CpGs out of another part of the genome, you get a further 10-fold amplification of replication. Very silly with hepatitis C as well. Con1 has been the gold standard replicon for hepatitis C research over the last 15 years. Hepatitis C is horrible to grow in a lab. In fact, there is only one isolate that will grow properly, but there are replicons. And again, these typically have neomycin, which is bacterial-derived, or luciferase as selection or reporter genes. And again, if you take the luciferase out and put a low CpG mutant in, you get this enormous enhancement of replication compared to the Con1 original sequence. These are really quite remarkable findings. Of course, it actually sort of has the implication as well that actually people are using this system where they're probably activating the cell all the time by an inappropriate sequence. So this has really quite important ramifications in terms of cell biology and immune response studies. The next part of the work was really to try and understand what pathways mediate these differences in replication ability of RNA viruses. And of course, there are many possibilities here. For example, are high CpG viruses more susceptible to interferon-stimulated genes? And we know that there are various nucleases like RNase L in the cell that are induced by interferon that might attack it more readily. Do the frequencies affect translation efficiency? And I won't show any data on that, but actually the answer is no, broadly. Actually, they probably do, but translation efficiency is not something that really governs replication ability of an RNA virus, at least in mammalian cells. The other more likely possibility is that they are more easily sensed by pattern recognition receptors. Many of you will know about MDA5 and RIG-I that actually recognize double-stranded RNA in the cytoplasm. Perhaps composition also influences that recognition process. There's also this very interesting protein called PKR, which it's also on one hand an ISG and will turn off translation when activated, but it's also involved in the early cellular stress response. And in most cell types, it's constitutively expressed and has a sort of target specificity that potentially encompasses single-stranded RNA virus genomes at least, and maybe that is in itself composition dependent as well. More exotically and less likely in terms of mammalian cells, siRNA or microRNA targeting. So these are all sort of things that we try to investigate, and I should say that a year and a half ago I wasn't an immunologist, and I still wouldn't be really confused with one. So this was quite a sort of struggle. I'm very grateful to people like Rick Randall who took me through many of the sort of very basic steps in this type of work. Looking at the far end, are high CpG viruses more susceptible to interferon? The answer is no. Okay, here's wild type. Here's the untreated control, and here's a massive dose of interferon, 500 units of interferon alpha per mil, and it knocks down Echo 7 replication by about tenfold, which is pretty resistant. But you'll notice that mutants, high CpG UpA, are actually very similarly affected, so that really doesn't account for their differences in replication ability. One thing we did notice that the replication defect occurs very early. If you do a synchronized experiment, you can look at RNA levels for ECHO7 within cells at 1 hour and very early time points. Post-infection, there seems to be no sort of defect in entry of high CpG mutants and high UpA mutants compared to the control, but the virus simply then fails to replicate. Okay, here's the wild type taking off at 4 hours, but in fact it's still stuck, showing perhaps only a 2 or 3-fold increase in the first 4 hours of infection. So that really does point very much towards recognition being a sort of key factor controlling the replication of these mutants. To try and investigate this, we did some pathway work, and this first experiment was interesting. This is work in cells expressing NPRO, which is a viral protein that turns off IRF3, which is really a key signaling molecule in the sort of pathway of MDA5 RIG-I and toll-like receptor 3. In fact, if you turn this very fundamental pathway off, ECHO7 doesn't really show much enhancement of replication, and that's actually because ECHO7 itself has evasion genes that completely destroy the initial sort of recognition system anyway. It actually is a protease that destroys MAVs. Okay, so the whole system is pretty dysfunctional in the enterovirus infected cells, and just turning off a lower part of the pathway results in a very modest increase in replication of the virus, and it's really very little affected in these high mutants as well. So we had a pretty good idea that the conventional pattern recognition receptors were not involved, and that's sort of underlined by these knockdown experiments. We had cell lines expressing small hairpin RNAs against MDA5, RIG-I, and PKR. And to a large extent, this is a linear scale, not a log scale. We saw very little difference in replication if we turned off any of these individual pattern recognition receptors. And I should point out particularly PKR, because it gets confusing later on. Well, I was certainly very confused in November last year. Again, concentrating a little bit on PKR, we did a second experiment with siRNA knockdown, and this was very efficient. We got greater than 80% reduction in protein expression in Western blots. And if anything, if you knocked PKR down, you actually knocked down the replication of the virus, whether it's wild type or mutant. So clearly, this is not a factor that's controlling the initial replication of the ECHO7 mutants, or indeed of the standard wild type virus. The reason we all got confused at Roslin was because we had also done some experiments with a chemical called C16, which traditionally is a specific inhibitor of PKR. It works in the micromolar range. It is minimally toxic and highly specific. And of course, the last two comments are really quite wrong, despite what the literature says. Just to visually show you what happens with C16, here's some monolayers of cells. We should explain to non-virologists, this is more or less a sort of intact monolayer of fibroblasts, and this is a virus, a monolayer showing a very strong cytopathic effect. So ECHO7 goes into cells and actually rounds them up, and eventually they all lyse. So we infect cells at a multiplicity infection of 0.1 and leave them for 24 hours. We get a full CpE of wild type. Whereas for the high mutants, it's very slow, and essentially these are essentially sort of normal monolayers without a CpE. If we do the same experiment again, but treat the cells with 2 micromolar of C16, we actually suddenly get a full CpE with the high mutants as well. Okay, so whatever seems to be inhibiting replication is suddenly de-repressed by this chemical inhibitor. And if we take the multiplicity down to 0.01, we're starting to see sort of much less change in the wild type, but we still see a full CpE with these high mutants. So looking at effects of this in a more quantitative way, if we look at the infectivity of virus stocks in the presence or absence of C16, we essentially see no difference with wild type virus. But we see a sort of 1,000-fold enhancement of replication of the CpG high mutant and about 100-fold of the high UpA. And essentially what this does is to restore their RNA infectivity ratios back to wild-type levels. So whatever fitness defect they have, it can be entirely reversed by pre-treating the cells with C16. We don't know how this works. We suspect there are other kinases and cells that are involved in RNA virus recognition that aren't characterized so far that are cross-inhibited by C16. So C16 isn't really perhaps very specific for PKR like many people think. And in fact, there are a huge number of unannotated kinases in the cell that are potentially involved in recognition that we'd like to biochemically characterize. We've done quite a lot of work in other systems, and I'll show you a very small amount of data on flu in a minute, but we can show the same sort of fitness enhancement and attenuation with Tyler's virus, which is another pecornus. And we think, given the universal suppression of CpG in RNA virus genomes, this seems to be a general attribute and a general pathway of recognition that controls the replication very widely across RNA viruses, at least those infecting mammals. We don't know whether whatever's selecting against CpG is also selecting against UpA, or are there different mechanisms involved in that? That's an interesting question to look at. And how does it work in plants? I mean, that's another sort of thing. Plants don't have interferon, so we really don't know how that really would work out. And then finally, why on earth haven't these viruses evolved spontaneously if they have this replication advantage? So clearly there's a fitness trade-off, and of course what we want to do is some proper mouse models where we can look at pathogenesis and some transmission rates. Just show you a very tiny bit of data that we're working on at the moment with flu A. We can actually make flu mutants where we take all CpGs or UpAs out of one or more segments, and I'll just show you some work with segment 5. Again, just the permuted controls, CpG, UpA high or low mutants. And at least with flu, we can infect mice with them, which is the problem with Echo 7. So with Paul Diggard and his group, we did a nice project where we infected mice at 200 PFUs, and this followed their sort of weight loss over time. We could then take their tissues out and actually look at viral loads and so on. So in general, the high mutants were highly attenuated in terms of pathogenicity, so they showed much less weight loss than wild type. And their viral loads in lungs were considerably lower than wild type or the CpG UpA low mutant that was done in the same time. When we looked at the pathology, we were very surprised that despite the low viral loads of high mutants, they induce a profound inflammatory response in the lungs. It was really quite different from wild type virus. So although this is a highly pathogenic virus, the inflammatory response is much greater in high mutants. So clearly whatever's recognizing this composition in the cell is actually translating through into the innate immune system and inflammatory response in the lung tissue itself. And remarkably, the pathology in the low mutants was actually somewhat less than the wild type if you scored it carefully. So it really does sort of beg the question of what we can do in terms of understanding why this sort of system doesn't sort of work more generally in terms of virus evolution. And clearly the key here are transmission experiments. So there are a number of sort of theoretical and practical issues arising from this, and this is going to be really the focus of my research over the next year or two. We really want to understand, whether these flu mutants, for example, or other model systems like Sendai virus and so on, which we can also set up, are affected in terms of transmission rate by having their composition altered, given that they seem to have no obvious in vivo defect that parallels that of the cell culture systems. The other sort of more practical issues arise around the fact that clearly all the vectors we've been using for mammalian systems, at least, could perhaps be redesigned, because clearly the insertion of luciferase, neomycin genes and so on, which we use conventionally for selection and reporter activities, are probably inappropriate in terms of the standard vertebrate cell system. We can also potentially enhance virus production to make better vaccines. Flu and poliovirus manufacture is limited by the amount of virus you can actually get to replicate in cells or eggs. We can probably up that by 10 or 50-fold by these low mutants. I think the other point is that high CpG mutants might be used as attenuated virus vaccines, but I think it's very important to understand the mechanisms by how that works. So, very finally, I'm not over time, am I? I'd like to thank very excellent postdoc in my group, Nicky Atkinson, who did really all the work on Echo 7. Jerome Vittel, a great postdoc in my lab for some time, who's been taking on some of those studies, and an Erasmus student, Ronnie Zhang, who's done work on flu. And the slides I showed on flu are a travesty of the progress he's made since I made those slides, and he's really got some great new data on the in vivo situation with these mutants. Helen Wise and Paul Diggard and his group, Pip Beard, the pathologist, Dave Evans, long-term collaborator, and Rick Randalls, whose hand held me through some innate immunity. Thank you.